Gene therapy is an experimental technique that uses genes to treat or prevent a disease. It involves introducing genetic material into cells to compensate for abnormal genes or to add a beneficial protein. For instance, if a mutated gene causes a protein to be faulty or even missing, it can disrupt normal development or cause a medical condition. Gene therapy may be able to supply a normal copy of the gene that can restore the function of the protein. But to deliver this gene to cells within the body, scientists need a delivery vehicle which they call a vector. It may be surprising, but certain viruses are often used as vectors.
Viruses are exquisitely well adapted to infecting their host. What's typically used in gene therapy are viruses that infect humans.
Usually when we think of viruses, we think of the ones that cause sickness, like the common cold, the flu, and HIV/AIDS. But scientists have been able to modify certain viruses to deliver selected DNA to cells in gene therapy without making people sick. Here's how it works. All viruses attack the cells of their hosts and inject their genetic material into the host cells. In gene therapy, viruses are engineered to carry good genes into a human cell and trigger the desired effect. The vector can either be injected directly into a specific tissue or organ in the body, or a sample of a patient's cells can be removed and exposed to the vector in a laboratory setting. Then the cells containing the vector are put back into the patient. If the gene does what it is supposed to, the treatment will be considered a success. In some cases, that means replacing the function of mutated genes, inactivating or knocking out a mutated gene that isn't working properly, or supplying genes that would help fight a disease like cancer, HIV, or cardiovascular disease. The first successful gene therapy patient was a 4-year-old girl suffering from a genetic disorder called ADA deficiency, which left her defenseless against infections. In 1990, doctors took some of her white blood cells, inserted normal ADA genes into them, and then re-injected the corrected cells. In a more recent example, researchers have successfully treated 2 boys suffering from a devastating brain disease called X-linked adrenoleukodystrophy, or ALD. They used a gene that was delivered by a virus derived from HIV. This marks the first time that a severe brain disease has been successfully treated by gene therapy. In less than 2 decades, gene therapy moved from the conceptual stage to laboratory research and onto clinical trials for a variety of life-threatening diseases such as hemophilia, cystic fibrosis, and neurodegenerative disorders.
The hope with gene therapy is that it provides a means for addressing some of these diseases that have been around for a long time. We now understand why they exist and how they work, and gene therapy offers an opportunity to fix what's broken.
Despite its promise, gene therapy can be risky. The viral vector may target the wrong cells, produce an infection, or may even cause a tumor to grow. The goal, however, is to make these treatments as safe as possible.